starting today, our lectures going forward will be on metabolism. So various metabolic pathways, how these biomolecules are utilized by our body. Either it's broken down to generate energy, or you use various precursors to synthesize biomolecules. Basically, from here onwards, we are going to jump straight into metabolic pathways. Like different types of reactions, enzymes, how energy is produced, oxidation reactions, reductive reactions, and so on. So starting with the metabolism, is on glycolysis. So the first metabolic pathway that we're going to do is metabolism of carbohydrates, and the first is glycolysis. So here, glucose is the main molecule that we're going to deal with. We're going to see how glucose is oxidized in the body in order to generate energy. Are you able to hear me? Or are you still waiting for the microphone? Sorry? You're waiting for the microphone?
Let me check.
Okay, good morning everyone. I hope you can hear me now. Yes? Okay, good. So yes, as I said, we'll be starting with metabolism, and the metabolism of carbohydrates with the pathway known as glycolysis. So in glycolysis, we are dealing with the biomolecule glucose. So before we look into the importance of glycolysis, just a brief introduction as to the importance of glucose itself. It's a carbohydrate, it is a monosaccharide, it is a hexose sugar, or more specifically, it is an aldohexose sugar, meaning it has an aldehyde functional group. It is a hexose sugar, meaning it has 6 carbon atoms. So here, glucose is very important to all animals or all living organisms because it is one of the most important metabolic fuels. So in our body, let's consider humans. For us, every cell in our body is capable of utilizing glucose as a metabolic fuel. So when I say utilize glucose, it means break down, or it means to oxidize glucose in order to generate energy. So whenever we talk about energy, we're talking about ATP molecules, because you know ATP molecule is the energy currency of the cell, isn't it? So when we say energy production, what we are meaning here is the production or the generation of ATP molecule. So glucose is very important in the glucose is very important because all the cells in our body are capable of oxidizing glucose in order to generate ATP molecules. We have other metabolic fuels or other molecules, biomolecules, which can act as metabolic fuels, such as fatty acids. And then there are ketone bodies, which you will be studying later on. So the difference between fatty acids and glucose is that Fatty acids can be utilized by only a certain number of tissues, meaning not all tissues can use fatty acids as a metabolic fuel. Example is your brain cells, your neurons, well, basically your nervous tissue, as well as your erythrocytes, that is the RBCs. These cells, they cannot utilize or they cannot break, they cannot oxidize fatty acid in order to generate energy. They solely depend on glucose. So that is why glucose is a very important molecule, because it is a metabolic fuel which can be utilized or which is used by all the cells in our body to generate ATP molecules or energy. So glucose, again, it's important because cells which can, under anaerobic condition, cells are able to still generate energy as long as you have glucose. So in the case of fatty acids, if fatty acids are the metabolic fuel, under anaerobic condition, fatty acids cannot generate energy. It needs an aerobic condition, meaning oxygen is required in order for fatty acids to be oxidized to generate energy. But in the case of glucose, even under anaerobic condition, I hope you know what anaerobic condition is, isn't it? In the absence of oxygen, or when the oxygen levels are low, glucose can still be oxidized to generate energy. So that again makes glucose an important and unique metabolic fuel, and for the fact that even under anaerobic condition, our body will be able to oxidize glucose to generate ATP molecule. Again, cells that lack mitochondria. Another difference. The oxidation of glucose takes place within the cytosol of the cell. So even in those cells that lack mitochondria, glucose is able to be oxidized to generate energy. Again, considering fatty acids, fatty acid oxidation takes place in the mitochondria. So in cells like the erythrocytes or RBCs, mature RBCs lack mitochondria. I hope you know that. Yes? So these cells that lack mitochondria, such as mature RBCs, even in these cells, glucose will be able to be oxidized to generate energy. Cells do not need mitochondria for energy production in the case of glucose when it is the metabolic fuel. But if you're using fatty acids as a metabolic fuel, the cells require mitochondria because the oxidation of fatty acid occurs within the mitochondria. Then tissues such as brains and RBCs, as I already mentioned, under normal condition cannot use any other metabolic fuel. So in the case of brains, your brain, in the absence or when the blood, when your the available glucose is very low, your brain can adapt. It can adapt itself to use other metabolic fuels such as ketone bodies. But the most preferred metabolic fuel for your brain is glucose. But if there is a complete lack of glucose, your brain can adapt itself to use another type of metabolic fuel known as ketone bodies. But even then, your brain cells cannot use fatty acids as a metabolic fuel because it lacks the enzymes required for oxidizing fatty acids. So again, that is why glucose is an important biomolecule, because it is the primary fuel or primary metabolic fuel in cells like the brains as well as the RBCs. And the reason for being so important is that glucose can be oxidized even under anaerobic condition to provide, to generate energy or ATP molecules. It can be oxidized even if the cells lack a mitochondria. So the concentration of glucose is, or the levels of glucose in the blood is regulated or controlled by a number of hormones, but the most important two hormones are peptide hormones known as glucagon and insulin. I'm assuming you've all heard of insulin. Yes? So insulin and glucagon, they're peptide hormones, meaning they're made up of amino acids, or they're made up of amino acids. So they're peptide hormones, and both these hormones are secreted by the pancreatic cells. So the pancreatic beta cells secrete insulin, whereas the pancreatic alpha cells secrete glucagon. The function of insulin is to lower the levels of glucose in the blood. So when your blood glucose level is high, insulin is secreted by the pancreas in order to lower it. Whereas the function of glucagon is to increase the levels of glucose in your blood. So usually glucagon is secreted when your blood glucose levels are low. So always remember this, insulin, its function is to lower blood glucose levels. So insulin will favor those metabolic pathways which utilizes glucose. Yes? The function of insulin is to lower blood glucose level. So insulin will favor all those metabolic pathways which utilizes glucose, meaning it will use up more and more glucose from the, or remove more and more glucose from the circulation, thereby bringing the blood glucose levels down. Whereas the function of glucagon is to increase the blood glucose level. So glucagon will favor those pathways or those metabolic pathways which increases or which synthesizes glucose. So you have your anabolic pathways and your catabolic pathways. So anabolic pathways are those that produce, synthesize, it's a biosynthetic pathway, isn't it? Whereas catabolic are oxidative or pathways that break down. So insulin will favor those pathways which utilize glucose, whereas glucagon will favor those pathways which synthesizes glucose. So these are the two main peptide hormones which play a role in regulating carbohydrate metabolism or maintaining the levels of glucose. So the overview of glycolysis. Already mentioned that glycolysis is an oxidative pathway. Here, glucose is the precursor, or glucose is the source of glycolysis. So, the glycolysis starts with glucose, where glucose undergoes oxidation. It can occur under aerobic as well as anaerobic conditions. So, glycolysis, even though it is an oxidative pathway, it can occur under both aerobic as well as anaerobic conditions. Glycolysis occurs within the cytosol of the cell. So the site of glycolysis is cytosol of the cell. Glycolysis is the first step of cellular respiration. So I'll talk more about the cellular respiration in the next slide. Now this pathway, it oxidizes glucose and splits it into two molecules of a 3-carbon compound known as pyruvate or pyruvic acid. As I already mentioned, glucose is a hexose sugar or a sugar containing 6 carbon atoms. So the 6-carbon glucose is oxidized and split into two molecules of a 3-carbon compound known as pyruvic acid or pyruvate. So a 6-carbon, it's unfortunate there is no whiteboard that I write on. So the 6-carbon glucose is split into two molecules of a 3-carbon compound known as pyruvic or pyruvate. So pyruvic acid or pyruvate is the end product of aerobic respiration or aerobic glycolysis. So pyruvate is the end product of glycolysis when there is adequate amount of oxygen or in cells that contain a mitochondria, because in cells that contain a mitochondria or when there is enough oxygen, glycolysis ends with pyruvate. Pyruvate now undergoes further oxidation in the mitochondria. So the end product of aerobic glycolysis is pyruvate. In cells that lack mitochondria, or in case there is, if glycolysis is occurring under an aerobic condition, then this pyruvate is further reduced into lactate or lactic acid. So the end product of aerobic glycolysis is pyruvate, or the end product of glycolysis in cells that contain mitochondria end product is pyruvate. So this pyruvate then is further oxidized in the mitochondria. Whereas in cells that lack a mitochondria, pyruvate is further reduced to lactic acid. Or if there is inadequate supply of oxygen, then pyruvate is converted to lactate. So the end product of anaerobic glycolysis is lactic acid, whereas the end product of aerobic glycolysis is pyruvate. So the conversion of glucose to lactic acid is known as anaerobic glycolysis because it occurs without the participation of oxygen. So even if oxygen is not available, lactic acid formation can occur. Anaerobic glycolysis, what is the importance of anaerobic glycolysis? So anaerobic glycolysis, it allows the cells to produce ATP even when there is no oxygen available. So this becomes especially important in cases such as hypoxia. So in our body, if the circulatory system suddenly is shut down or momentarily is shut down, the anaerobic glycolysis allows our cells to produce ATP molecule in order to maintain the basic cellular function. Yeah, because if this wasn't the case, even for a split second, the oxygen supply to your body, to your brain is cut off, that means your cells will start dying, isn't it? Because there's no oxygen for energy production. But anaerobic glycolysis, it sort of helps your body to continue producing ATP molecule even in the absence of oxygen. So in cases such as hypoxia, that's when the there's low oxygen supply to the various tissues. So even in such cases, your body can still continue producing energy, ATP molecules, by carrying out anaerobic glycolysis. So if your body was dependent on, say, fatty acids for its energy needs, what would happen is that in cases like hypoxia, fatty acid oxidation cannot occur because fatty acid oxidation only occurs in the presence of oxygen. So if oxygen is cut off, then fatty acid oxidation does not take place and no ATP is formed. And your cells, they require a constant supply of ATP in order to carry out the various cellular functions. Clear? Yes? So that is why anaerobic glycolysis is very important, in that even if your body or your tissue certain part of your tissue does not get enough oxygen, it is okay. It does not die immediately because the tissue will be able to carry out anaerobic glycolysis to produce ATP molecules. And this ATP molecules will help it carry out its cellular function. Yes? So, cellular respiration. Cellular respiration, it is a biochemical term. So you have respiration and you have breathing. So breathing is a mechanical process. So that's like where you use your muscles, your diaphragm, in order to inhale air and inhale oxygen and exhale carbon dioxide. Yes? So breathing, it is a mechanical process, whereas respiration, it is a biochemical process. It involves these three pathways together constitutes respiration. So respiration is a biochemical process which involves glucose, oxygen to produce carbon dioxide, water and energy. So cellular respiration has three stages. Stage 1 is glycolysis, which occurs in the cytosol. Stage 2 is the citric acid cycle, which takes place in the mitochondria. And stage 3 is the electron transport chain or the oxidative phosphorylation, which also occurs within the mitochondria. So these three stages together constitute cellular respiration. So I hope you're familiar with the chemical formula: glucose plus oxygen giving carbon dioxide plus water plus energy. Yes? Direct, yes? So your whole cellular respiration can be summarized by that formula, that is glucose, or C6H12O6 plus O2 gives rise to carbon dioxide, or CO2 plus H2O plus energy. So that chemical equation summarizes the 3 stages of cellular respiration. The glucose enters into stage 1, that is glycolysis, and the products carbon dioxide, water and energy are produced during the 3 stages. So as you can see here, during stage 1 there is ATP production, or energy production. The end product of glycolysis, which I mentioned, is pyruvate. In case of aerobic respiration, I mentioned that the pyruvate enters into the mitochondria to be further oxidized. So here again, pyruvate enters into the mitochondria to be further oxidized, and during that process carbon dioxide is produced as well as ATP molecules. Now, because these stage 1 and stage 2 are oxidative processes, so you know what oxidation means, isn't it? There's removal of electrons. Yes? Yes? Oxidation means there is removal of electrons. So when these electrons are removed, you need electron carriers to accept it. So the electron carrier here is NADH and FADH2. So in glycolysis, the electron carrier is NADH. And in stage 2, that is the citric acid cycle, you will see that NADH and FADH2 are also produced. So most of the energy that is liberated during these oxidative pathways are stored in NADH and FADH2. So the NADH and FADH2 from stage 1 and stage 2, that is from glycolysis and citric acid cycle, are further channeled into oxidative phosphorylation, where the energy stored in NADH and FADH2 are used to produce ATP molecules. So during these process, carbon dioxide is produced during the citric acid cycle, and during oxidative phosphorylation, water molecules and energy, or ATP molecule, is produced. So this carbon dioxide and water molecule that is produced in stage 2 and 3 are removed from your body when you exhale. Here. So anyway, that's just like what cellular respiration is, or the 3. Because if someone asks you what is cellular respiration, you need to say that it's a biochemical process which involves glycolysis, citric acid cycle and oxidative phosphorylation to generate energy. Also, you have carbon dioxide and water, which are the byproducts. So today we are going to focus on stage 1, that is glycolysis, where glucose is utilized, or where glucose is oxidized. So before glycolysis can occur, glucose needs to be taken up, or it needs to enter into the cells in order for it to undergo glycolysis. So the plasma membrane of cells, they are impermeable to glucose. So meaning glucose cannot freely diffuse across the plasma membrane. They require specialized transporter proteins that will carry the glucose from outside the cell into the cell. And you have two transport mechanism. You have a sodium independent facilitated diffusion transport. So this system is mediated by a family of glucose transporters which are present on the plasma membrane, and these glucose transporters are known as GLUT, or GLUT, that stands for glucose transporters. So there are different types of glucose transporters. So in each tissue you will find a particular type of glucose transporters, and they are specified using numbers. So as you can see here, GLUT1, it is present in RBCs, in the blood-brain barrier, as well as in the blood-placental barrier. Then you have GLUT2, which is found in hepatocytes, or the liver cells, in kidney cells, in the pancreatic cells, in the pancreatic beta cells, as well as in the gastrointestinal tract. Then you have GLUT3, which is found in the brain and the neurons. Basically, GLUT3 are found in the nervous tissues. So GLUT1, GLUT2, GLUT3, they are embedded in the plasma membrane. So as you can see here, So as you can see here, this yellow structure here is the glucose transporters. So GLUT1, GLUT2, GLUT3, these glucose transporters, they are embedded in the plasma membrane of the respective cells. So as these gray color structures are glucose molecules. So when the glucose molecule binds to the GLUT protein that is embedded in the plasma membrane, they will close. So once glucose binds to the GLUT transporter, it will close and it undergoes a conformational changes which brings about. So here in the first conformation, it is open towards the outside. So as you can see here, the GLUT is open towards the outside of the cell so that the glucose that is present in the blood can bind to the GLUT. Once it binds to the GLUT, it closes. As you can see here, it has closed and it undergoes certain conformational changes resulting in the opening of the GLUT transporter towards the inside of the cell. So when it opens towards the inside of the cell, it now pushes the glucose that it was bound to into the cells. Is this clear? Yes? So the GLUT 1, 2, 3, they have two conformation, an open conformation and a closed conformation. So initially, the open conformation, the opening is towards the outside of the cell. Once glucose binds to the GLUT protein, it closes up. So as you can see here, as the arrow indicates, it has closed up and it undergoes a conformational change where it opens towards the inside of the cell, and by opening into the cell, the glucose can now enter into the cell. Yes? So GLUT 1, 2 and 3, these glucose transporters, they are permanently embedded in the plasma membrane of the respective tissues. So meaning they are always present on the plasma membrane. Now GLUT2, it is different from GLUT 1 and 3 in that it allows for bidirectional transport of glucose. So meaning this same GLUT2 can allow the transport of glucose from outside the cell into the cell, as well as the transport of glucose from inside the cell to the outside. So meaning the transport of glucose occurs bidirectional. Yes? Whereas GLUT 1 and GLUT 3, the direction of glucose transport is unidirectional, meaning it only allows the transport of glucose from outside the cell into the cell. Clear? Yes? GLUT 1 and GLUT 3 only allows the entry of glucose into the cell. It does not allow glucose to move out of the cell. Clear? Whereas GLUT 2, it allows the glucose to enter the cell as well as exit the cell. Clear? Yeah. Next you have GLUT4, which is found in adipose tissues as well as skeletal muscles. So as you can see here, this GLUT4 is also different from the other glucose transporters that I mentioned, in that they are internalized. So here as you can see, this is the cell. The GLUT4, it is bound to a vesicle and it's present within the cytosol of the cell. So when insulin is secreted, so I said when your blood glucose levels are high, insulin is secreted. This insulin will bind to its receptor. The binding of insulin to the receptor will trigger the movement of the GLUT4, which is in the cytosol, to move towards the plasma membrane. So here, as you can see, in the presence of insulin, the GLUT4 vesicle, it fuses with the plasma membrane. So once the GLUT4 vesicle fuses with the plasma membrane, now it allows for glucose to enter into the cell. Clear? So here, GLUT4, the activity of GLUT4 is dependent on the presence of insulin. So meaning if insulin is absent, then GLUT4 will remain internalized or will remain within the cytosol. Yes? So, yes? If insulin is absent, if insulin is not secreted, then GLUT4 will remain internalized, or it will remain within the cytosol. If GLUT4 remains within the cytosol, then glucose cannot enter into these cells, which is the adipocytes and the skeletal muscles. Clear? Yes? So that is what happens in diabetes mellitus. In diabetes, there is a deficiency of insulin, isn't it? Yes? I'm hoping you know that diabetes is caused due to deficiency of insulin, or there is deficiency of insulin. Yes? And in diabetes, because there is deficiency of insulin, there is also hyperglycemia, or abnormally high blood glucose level. The reason for the abnormal high glucose level is because GLUT4, as I mentioned, remains internalized because there is no insulin present, meaning glucose cannot enter into the adipocytes as well as the skeletal muscles. Instead, they remain in the circulation. Clear? Yes? So GLUT4, it requires insulin for it to be activated, or for it to move to the plasma membrane, so that glucose can be taken up by adipocytes as well as skeletal muscles. Whereas in the case of GLUT1, 2 and 3, it does not matter if insulin is present or absent, because they are permanently embedded in the plasma membrane. Clear? It's only GLUT4 that requires insulin for it to move from the cytosol to the plasma membrane. And only once it fuses to the plasma membrane can it allow the uptake of glucose into the cell. So the number of GLUT4 transporter active in the tissue is increased by insulin. So as insulin is secreted, the number of glucose transporters, or GLUT4, that is activated is also increased. Insulin, it promotes the transfer or translocation of GLUT4 from the cytosol to the plasma membrane. And once it fuses with the plasma membrane, it can now allow the uptake of glucose into the cell. The next type of mechanism is sodium monosaccharide co-transporter system. So in this system, both glucose and sodium ions are transported across the, yes. It's unidirectional. So the only GLUT transporter protein that allows bidirectional transport of glucose is GLUT2. GLUT1, GLUT3 and GLUT4, they are all unidirectional. They only allow the entry of glucose. It is only GLUT2 that allows for glucose to move out of the cell. Yes? Sorry. So yes, so in this sodium monosaccharide co-transporter system, both sodium and glucose are transported across the plasma membrane by a transporter protein known as SGLT, or sodium glucose transport molecule. So this type of transport molecule is found in the epithelial cells of the enterocytes, or the intestinal cells, as well as in the renal tubules, or in the, to be more precise, in the proximal tubules of nephrons. Now we move on to glycolysis. So glycolysis, it is an oxidative pathway. It is made up of 10 reactions. So as you can see here, first you have glucose. It undergoes 5 reactions, and it is split into 2 3-carbon compounds. So the glycolysis, the entire glycolytic pathway, it can be split into 2 phases. The first phase is known as preparatory phase. So the first 5 reactions of glycolysis is known as preparatory phase. This is because during the preparatory phase, there is need for 2 ATP molecules. In order for the oxidation reaction to occur, you need to invest in 2 ATP molecules. So the first stage, the first phase of glycolysis is known as preparatory phase. So preparatory phase is made up of the first 5 reactions, and it involves the investment or the participation of 2 molecules of ATP molecules. The last 5 reactions of glycolysis is known as the payoff phase. It is during the payoff phase where there is production of ATP molecules. So in the preparatory phase, ATP molecules are utilized. In the payoff phase, ATP molecules are generated. Clear? So the first 5 reactions of glycolysis is known as preparatory phase, where there is utilization of ATP molecule. The last 5 reactions of glycolysis is known as payoff phase, and it involves the generation or it involves the production of ATP molecules. So apart from ATP molecule, energy is also stored in the form of NADH. So it's not just ATP that is formed during payoff phase, but also NADH molecule. So let's begin. Just point out that you do not need to know the structures of the intermediates. You just need to know the reaction and the enzymes that are involved, as well as the activators and inhibitors. You do not need to know the structure. As I already mentioned, you don't need to know the structure of the compounds. You just need to know the name and the enzymes reaction. So the first reaction is the phosphorylation of glucose. So this is an important reaction because once glucose enters into the cell, phosphorylating it will basically trap the glucose in the cell. This is especially important in the case of liver, kidney cells because they contain transporter molecules, isn't it? Yes? And glucose transport molecules, they are involved in bidirectional transport. So if glucose, as soon as it enters into the cell, if you do not trap it, it can move back using the same transporter molecule, isn't it? Yes? So that phosphorylation of glucose becomes important. So as soon as glucose enters into the cell, it is phosphorylated by the enzyme hexokinase to form glucose 6-phosphate. So here a phosphate group is donated by ATP molecule. So here is the first ATP molecule that is utilized. ATP molecule donates one of its phosphate groups to glucose to give you glucose 6-phosphate. So glucose 6-phosphate, the 6, it represents the carbon atom at which the phosphate molecule is attached to. So glucose 6-phosphate means the phosphate group is attached to C6 of glucose. Clear? So here the number represents the position of the functional group. So when I say glucose 6-phosphate, it means that the phosphate group is attached to C6 of glucose. And here, once glucose is phosphorylated, it is pretty much trapped. The glucose is trapped in the cell. It cannot move out of the cell because there is no transporter molecule that can transport phosphorylated glucose. So this pretty much makes sure that the glucose is trapped within the cell. So this reaction, the phosphorylation of glucose to glucose 6-phosphate, it is an irreversible reaction. So this first reaction of glycolysis, it is an irreversible reaction. Hexokinase, it is inhibited by its product, which is glucose 6-phosphate. So high levels of glucose 6-phosphate can inhibit hexokinase, whereas it is activated by high levels of AMP and ADP. Now in liver cells as well as the pancreatic beta cells, there is an enzyme known as glucokinase. So in the hepatic cells and in the pancreatic beta cells, glucokinase is responsible for phosphorylating glucose to glucose 6-phosphate. Clear? Hexokinase, it phosphorylates all hexose sugar, meaning it's not just glucose, it will also phosphorylate fructose, galactose and so on because they are all hexose sugar. Clear? So hexokinase is specific for hexose sugar, whereas glucokinase, it is specific for glucose. Clear? So hexokinase, that enzyme is present in all the cells in the body, whereas glucokinase is only present in the liver cells as well as the pancreatic beta cells. So in the liver cells and the pancreatic beta cells, Glucokinase is responsible for phosphorylating glucose to glucose 6 phosphate. And here, glucokinase is inhibited by fructose 6 phosphate. Clear? So since they are different enzymes, they are also inhibited by different molecules. Hexokinase is inhibited by glucose 6 phosphate, whereas it is activated by AMP and ADP. Glucokinase, it is inhibited by fructose 6 phosphate. So this is the first reaction. The second reaction is the isomerization of glucose to fructose. I hope you know that glucose and fructose, they are isomers of each other. Yes? Meaning they are made up of the same number of carbon, hydrogen and oxygen atom. They have the same chemical formula, isn't it? C6H12O6. It's just that they are arranged in different ways. So here, in this reaction, glucose is isomerized to fructose. So glucose 6-phosphate is isomerized to fructose 6-phosphate by the enzyme phosphohexose isomerase, and this reaction is reversible, meaning the same enzyme can catalyze the reaction from glucose 6-phosphate to fructose 6-phosphate and fructose 6-phosphate to glucose 6-phosphate. Clear? I hope this reaction is pretty simple. So the second reaction is the isomerization, meaning conversion of glucose 6-phosphate to fructose 6-phosphate by the enzyme phosphohexose isomerase. This reaction is reversible. The first reaction was irreversible and this second reaction is reversible. Please take note of all those reactions which are irreversible, because these reactions are also regulatory reactions, meaning you control the rate of a pathway by regulating those reactions. So the first reaction was irreversible. The second reaction is reversible. Third reaction is also irreversible. Here again, the fructose 6-phosphate, it undergoes another phosphorylation reaction, meaning the addition of a second phosphate group. So here, the second ATP molecule is utilized. So as you can see here, fructose 6-phosphate to fructose 6-phosphate, another phosphate group is donated by ATP molecule to give you fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate means you have phosphate groups attached at C1 position and C6 position of fructose. Clear? This phosphorylation reaction is also irreversible. So the PFK1, that is phosphofructokinase 1, that is the enzyme which catalyzes this phosphorylation reaction. So PFK1 reaction is irreversible under cellular condition and it is the first committed reaction. So meaning once this reaction occurs, then all the reactions of glycolysis will take place. So hence it's known as the first committed reaction or first committed step. Now PFK1 or phosphofructokinase 1, it is inhibited by high levels of ATP and citrate, and it is activated by high levels of AMP. It is also activated by a compound known as fructose 2,6-bisphosphate. So please bear in mind, fructose 2,6-bisphosphate is different from fructose 1,6-bisphosphate. Clear? They are two different compounds. Yes? So if I ask you what is the activator of fructose, what is one of the activators of phosphofructokinase 1, please do not write fructose 1,6-bisphosphate. That will be wrong. Clear? It is fructose 2,6-bisphosphate. Yes? Yes? So don't get, it's just a number different, but there are two different types of molecules. So phosphofructokinase 1 is activated by high levels of AMP as well as high levels of fructose 2,6-bisphosphate, and it is inhibited by high levels of ATP and citrate. So this is the second irreversible reaction, which is the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. Again, second molecule of ATP will donate its phosphate group. And here the phosphate groups are attached at C1 and C6 position. The fourth reaction is the splitting of fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate is a 6-carbon containing compound. It is now split into two 3-carbon containing compounds, which is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Each of this have 3-carbon compounds. Clear? They are triose sugar. Yes? Yes? So fructose 1,6-bisphosphate, it is split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and the enzyme responsible for this is known as aldolase. This again is a reversible reaction. So as you can see here from the arrow, it is a reversible reaction. So the 6-carbon fructose 1,6-bisphosphate is split into two molecules of 3-carbon dihydroxyacetone phosphate and 3-carbon glyceraldehyde 3-phosphate. Now, only glyceraldehyde 3-phosphate can continue in the glycolytic pathway. Dihydroxyacetone phosphate, it cannot continue in the glycolytic pathway unless it is converted to glyceraldehyde 3-phosphate. So here, both of these compounds, they are isomers of each other. Just like how glucose and fructose are isomers of each other, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they are isomers of each other, meaning they have the same chemical formula, but they are structurally different. So in order for dihydroxyacetone phosphate to continue in glycolytic pathway, it is converted into glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. So the enzyme triose phosphate isomerase will convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. And this is reaction 5, which is the last reaction in the preparatory phase. So at the end of reaction 5, you will have two molecules of glyceraldehyde 3-phosphate. Clear? At the end of reaction 5, you will have two molecules of glyceraldehyde 3-phosphate. Clear? Yes? So from now onwards, from the next reaction onwards, the reactions will take place twice because you have two molecules of glyceraldehyde 3-phosphate. Yes? Is that clear? So this reaction is also reversible. Conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, it is a reversible reaction. So reaction 6 onwards, the payoff phase starts. So reaction 6 is the beginning of the payoff phase. So this is the phase where ATP molecules are produced or generated. So far we had ATP molecules that were utilized. We had an ATP molecule that was utilized in the first reaction, phosphorylation of glucose. Then you had another ATP molecule which was utilized during the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. Starting from here, now will be the energy production phase. So reaction 6 is the oxidation of glyceraldehyde 3-phosphate. So please pay attention from reaction 6 onwards. All the reactions will occur twice because you have two glyceraldehyde 3-phosphate molecules. So all the reactions starting from reaction 6 will occur 2 times. So here glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate. So during this oxidation reaction, an inorganic phosphate molecule is also attached to glyceraldehyde 3-phosphate. And because it is an oxidation reaction, there is the removal of electrons. Clear? Whenever you are told that a reaction is oxidation, that means there is going to be removal of electrons. And in all oxidation reactions, you require an electron carrier. The electron carrier can either be NAD+, FAD or NADP. Clear? So these are the 3 types of electron carriers, either NAD+, FAD or NADP. So these electron carriers will accept the electrons and they become reduced. Clear? Is said to be oxidized. When a compound gains electrons, it's said to be reduced. So please don't forget that. So here, because it is an oxidation reaction, you have an electron carrier which is NAD plus. So here NAD plus is the electron carrier. It will accept the electrons that is removed from glyceraldehyde 3-phosphate. And to glyceraldehyde 3-phosphate, a molecule of inorganic phosphate is also attached to form 1,3-bis phosphoglycerate. So again, 1,3-bis phosphoglycerate, meaning the phosphate groups are attached at C1 and C3 position. So as you can see here, this is C1, C2 and C3. So here you have a phosphate group at C1 position and another phosphate group at C3 position. This oxidation reaction is also a reversible reaction and it is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Yes? So reaction 6 is the oxidation of glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate. Here, because it is an oxidation reaction, NAD plus is reduced to NADH. It accepts the electron and it gets reduced to NADH. It is a reversible reaction. You also require a inorganic phosphate to form 1,3-bis phosphoglycerate. So this is the first oxidation reduction reaction. It is an oxidation reduction reaction because glycol is oxidized whereas NAD plus is reduced. Hence, it's known as an oxidation reduction reaction. Clear? Yes? Now, some clinical significance of this enzyme, which is glyceraldehyde 3-phosphate dehydrogenase. This enzyme, it can be inhibited by hyperglycemia. So that is where there is abnormally high blood glucose level. Here, hyperglycemia is abnormally high blood glucose level, whereas hypoglycemia is abnormally low blood glucose level. Clear? So that's hyper, H-Y-P-E-R. Hyperglycemia is abnormally high blood glucose level, whereas hypo, that's H-Y-P-O, hypoglycemia is abnormally low blood glucose level. So here, under hyperglycemic condition, the enzyme glyceraldehyde 3-phosphate dehydrogenase is inhibited. So this inhibition is brought about by an activation of an enzyme known as poly-ADP ribose polymerase. So this enzyme, poly-ADP ribose polymerase enzyme, is activated in hyperglycemia. This enzyme, what it will do is, it will add multiple copies of ADP ribose molecules to the cysteine sulfhydryl groups that is present in the active site of glyceraldehyde 3-phosphate dehydrogenase. So you've already done enzyme, in that you know how important the active site of enzymes are, isn't it? Yes? The active site is where the substrate binds and the catalytic action takes place, isn't it? Yes? And there are certain amino acid residues that are present in the active site which are essential for the function of the enzyme. Clear? So in the case of glyceraldehyde 3-phosphate dehydrogenase, cysteine sulfhydryl groups, you know the amino acid cysteine, yes? Sulfhydryl group, it is a functional group, that is SH. So cysteine amino acids, they have these sulfhydryl groups, that is the SH group. This sulfhydryl groups that are present in the active site of the enzyme is essential for the enzyme activity. If this sulfhydryl group is bound by any molecule, then the active site is unavailable to be bound by the substrate. Clear? In order for the substrate to bind to the active site, you need the cysteine sulfhydryl groups. Clear? It needs to be free. Yes? But the poly-ADP ribose polymerase enzyme, it will add multiple copies of ADP ribose to the free sulfhydryl groups in the active site. So because these free sulfhydryl groups have now multiple copies of ADP ribose bound to it, the enzyme activity is inhibited. Clear? Now, substrate cannot bind to the active site of the enzyme. Is that clear? That brings about the inhibition of the enzyme. So, hyperglycemia can bring about inhibition of glyceraldehyde 3-phosphate dehydrogenase, meaning the payoff phase of glycolysis is inhibited because it is the first reaction of payoff phase, isn't it? So if this enzyme is inhibited, meaning the payoff phase of glycolysis does not take place. Reaction 7 is the synthesis of 3-phosphoglycerate as well as the formation of a ATP molecule. So here 1,3-bisphosphoglycerate, it donates one of its phosphate group to ADP. So here it's pretty much one of the phosphate groups from 1,3-bisphosphoglycerate is donated to ADP to form ATP and 3-phosphoglycerate. So here 1,3-bisphosphoglycerate, the phosphate group at C1 position is donated to ADP to give you ATP and 3-phosphoglycerate. So the first molecule of ATP is produced in reaction 7. Clear? Yes? And since this reaction takes place twice, you have 2 molecules of ATP that is formed. Clear? And this reaction is such a reaction where the substrate donates its phosphate group to ADP to form ATP is known as substrate level phosphorylation. Clear? Yes? So here the substrate 1,3-bisphosphoglycerate donates its phosphate group to ADP to form ATP molecule. Is that clear? I hope that's pretty straightforward. The phosphate group at C1 position is transferred to ADP to form ATP. Yes? And this type of reaction is known as substrate level phosphorylation. This reaction is also reversible. So this is also a reversible reaction. And the enzyme responsible for the substrate level phosphorylation is known as phosphoglycerate kinase. So phosphoglycerate kinase is responsible for the formation of a ATP molecule. So substrate level phosphorylation, it is a mechanism of ATP formation where the substrate donates its phosphate group to ADP to form ATP. So it's just so as you can see here, you have the enzyme here, the substrate with a phosphate group and ADP. So the enzyme catalyzes the transfer of the phosphate group from the substrate to ADP to form ATP. So this is known as substrate level phosphorylation. In the case of arsenic poisoning, what happens is arsenic, it bypasses, it's unfortunate.
It's going behind.
Sorry? Yep. Can someone help me move the... Sorry? It won't move. No, but in the sense, turn it around. That's what I meant. Is there any way for you to source one code? Because it's easier to explain when... So you have... Okay, so here, this is the reaction 6, that is glyceraldehyde 3-phosphate is oxidized to 1,3-bisphospho. Glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate with the formation of NADH. Once 1,3-bisphosphoglycerate is formed, it undergoes substrate level phosphorylation to form 3-phosphoglycerate and ATP molecule. Yes? So that's reaction 6 and 7 that we already discussed. Yep. So in arsenic poisoning, what happens is arsenic, it binds to glyceraldehyde 3-phosphate dehydrogenase enzyme and it bypasses this reaction. So meaning in presence of arsenic, glyceraldehyde 3-phosphate, it hydrolyzes or it is oxidized to 3-phosphoglycerate. And so it gets oxidized to 3-phosphoglycerate, meaning these two reactions do not occur. Clear? Yes? Arsenic, in the presence of arsenic, arsenic binds to glyceraldehyde 3-phosphate dehydrogenase enzyme and it bypasses the formation of 1,3-bisphosphoglycerate. So meaning glyceraldehyde 3-phosphate, it is oxidized or it is converted to 3-phosphoglycerate. So these two reactions don't take place, which means that ATP molecule production is, there is no ATP production, isn't it? Because here glyceraldehyde 3-phosphate, it just gets converted to 3-phosphoglycerate and glycolysis occurs, whereas the formation of NADH and ATP is bypassed. So there is no production of NADH and ATP. Clear? So the energy production in arsenic poisoning as a result of glycolysis is reduced. Clear? Yes? So thus there will be no net ATP production occurring in the presence of arsenic because arsenic will bypass the formation of 1,3-bisphosphoglycerate and ATP. So in presence of arsenic, glyceraldehyde 3-phosphate is converted to 3-phosphoglycerate without the formation of NADH and ATP molecules. Clear? So the energy production becomes low in the presence of arsenic. Yes? So once 3-phosphoglycerate is formed, it undergoes another isomerization reaction. So basically here, it is just the transfer of phosphate group. So here, in 3-phosphoglycerate, the phosphate group is attached at C3 position. In presence of the enzyme phosphoglycerate mutase, the phosphate group is moved to the C2 position. So it becomes 2-phosphoglycerate. So this again is a reversible reaction. So reaction 6, reaction 7 and reaction 8, these are all reversible reactions. So here phosphoglycerate mutase will transfer the phosphate group from C3 position. As you can see here, the phosphate is in the C3 position. So from the C3 position, it will be transferred to the C2 position to form 2-phosphoglycerate. Next is the dehydration reaction. So dehydration reaction meaning a removal of a water molecule. So from 2-phosphoglycerate, a water molecule is removed to form phosphoenol pyruvate. And this dehydration reaction is catalyzed by the enzyme enolase. So as you can see here, a hydrogen atom from C2 position and a hydroxyl group from C3 position is removed as a water molecule and a double bond is formed between C2 and C3 position to form phosphoenol pyruvate. This reaction is also a reversible reaction. Now we come to the final reaction, the last reaction of glycolysis, which is the formation of pyruvate. So here this is again a substrate level phosphorylation. The phosphate group attached to phosphoenol pyruvate, it's donated to a ADP molecule to form ATP, and you have a pyruvate molecule. So the enzyme responsible for this reaction is known as pyruvate kinase. Sorry about that. So the enzyme that catalyzes this reaction is known as pyruvate kinase. This last reaction is an irreversible reaction. So the phosphate group attached at C, the phosphate group attached to phosphoenol pyruvate is donated to ADP to form ATP. So the products of the last reaction is a molecule of pyruvate and a molecule of ATP. Clear? Now, in liver, pyruvate kinase is activated by fructose 1,6-bis phosphate. So, the activator of pyruvate kinase is fructose 1,6-bis phosphate. The enzyme glucagon, it indirectly inhibits pyruvate kinase activity. So, if the levels of glucagon are high in the system, it will inhibit pyruvate kinase activity so that the last reaction of glycolysis does not occur. So at the end of glycolysis, so at the end of glycolysis, you have glucose, which gives you 2 molecules of pyruvate. So you have 2 molecules of pyruvate along with 1 molecule. So here during this reaction 6, you have 1 molecule of NADH. Reaction 7 gives you an ATP molecule, so reaction 6 gives you 1 molecule of NADH. Reaction 7 gives you a molecule of ATP, and the last reaction, reaction 10, also gives you an ATP molecule. Similarly, here also. So at the end of glycolysis, from 1 molecule of glucose, you get 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. So the products of aerobic glycolysis is 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. Clear? Yes? But we say that there is a net gain of only 2 ATP molecules, because during the preparatory phase we used up 2 molecules of ATP, isn't it? Yes? So we're going to take away the 2 molecules from here because we use that, let's just say it was like a loan. Yes? From your cells reserve, you use 2 ATP molecules in order to start glycolysis. Yes? So once you produce the ATP molecule, now you need to return those 2 molecules. Yes? So you only have a net gain of 2 ATP. I hope that's clear. Yes? Okay. Now, a clinical significance with regard to glycolysis is a deficiency in pyruvate kinase. So the deficiency of pyruvate kinase, it is yes. So basically these magnesium and ions, they are cofactors. They are needed in order for the enzyme to be functional. So whenever you see magnesium ion, it means that magnesium ion is required for the enzyme to be fully functional. Haven't you learned about cofactors and coenzymes? Huh? So some of you have and some of you haven't. So there's these cofactors and coenzymes. These are prosthetic groups, or they are molecules that are required by the enzyme in order to be fully functional. So here the magnesium ions are cofactors. It is required in order for the enzymes to be fully functional. A deficiency of magnesium ion can mean that the functionality of the enzyme is reduced here. So basically magnesium ion is required for the function of the enzyme. Yes, so as I was saying, pyruvate kinase deficiency. So here it affects the pyruvate kinase deficiency. It affects RBCs, because RBCs, they require or they depend on glycolysis, and or glycolysis is their major energy producing pathway. So here the normal mature erythrocytes, it lacks mitochondria, so it completely depends on glycolysis for its ATP production. Now the ATP that is produced by glycolysis is required for the metabolic needs of the RBCs, as well as for maintaining the RBCs. So it helps in maintaining the biconcave and flexible shape of the cells, which allows the RBCs to squeeze through thin capillaries. Now, when the ATP production or glycolysis is inhibited, if there is not enough ATP production, then the cellular structure of RBCs become susceptible, or it can be easily ruptured. So it causes hemolysis. Clear? So RBCs, they need a constant supply of ATP in order to maintain the structural integrity of the cell. Clear? Yes? ATP is required by RBCs in order to maintain their structural integrity. So if the pyruvate kinase, in case of pyruvate kinase deficiency, the last step, that is step 10, does not occur, meaning 2 molecules of ATP are not produced, isn't it? So there is no net gain of ATP, meaning there is no ATP production. If there is no ATP production, then the structure of the RBCs become weak, and as they try to squeeze through capillaries, they will be easily ruptured. They will undergo lysis or cell death. Clear? Causing hemolysis. So more and more RBCs will undergo premature hemolysis, causing anemia. Is that clear? Because more and more RBCs are dying or undergoing hemolysis, so meaning the oxygen carrying capacity of the RBCs are reduced, causing anemia. Clear? So symptoms of pyruvate kinase deficiency is mild to severe, causing chronic hemolytic anemia, icterus, fatigue, jaundice, and elevated bilirubin levels. So all these other symptoms, such as jaundice and elevated levels of bilirubin, you will know its correlation to hemolytic anemia when you're doing heme metabolism. You'll see how, as more and more RBCs undergo lysis, there is increased catabolism of hemoglobin, resulting in high levels of bilirubin. So all you need to know is that ATP, a constant supply of ATP is required by RBCs in order to maintain their structural integrity. If there is no ATP production, then the cellular structure of RBCs becomes very fragile and they undergo lysis, leading to hemolytic anemia. Yes? So in the case of aerobic glycolysis, the end product is pyruvate. Now pyruvate, when there is adequate amount of oxygen and also in cells with mitochondria, the pyruvate enters into the mitochondria to be further oxidized in the citric acid cycle. which is stage 2 of cellular respiration. But in the case of cells that lack mitochondria, or in the presence of, or in the absence of oxygen, what happens to this pyruvate? As I already mentioned, the end product of anaerobic glycolysis is lactate, isn't it? So pyruvate will be further reduced to lactate. Now, because there is only a limited amount of NAD plus available, the NADH needs to be recycled. So, as you know, in reaction 6, NAD plus is converted to NADH, yes? So, the amount of NAD plus available in the cell is very limited. Clear? So, as more and more NAD plus is converted to NADH, in order for glycolysis to continue occurring, you need NADH to be converted back to NAD plus. Yes? In this reaction, place you need NAD plus, isn't it? You need an electron carrier. Yes? If NAD plus is not available, this reaction will not take place because glyceraldehyde 3-phosphate needs to donate its electron to someone. Yes? What if all the NAD plus in your cell has already been converted to NADH? Then what happens? This reaction will not take place, isn't it? If there's no NAD plus available, this reaction will not take place. Yes or no? Yes. So now you need the reaction to continue occurring because you need ATP production to occur. Yes? So what is the solution to it? The solution is to convert the NADH back to NAD plus. Isn't it? Yes or no? I mean, that makes, that's the only logical sense, isn't it? If you only have 10 NAD plus, all the 10 NAD plus has been converted to NADH. Yes? Now glycolysis will not occur because there's no NAD plus. What do? You have to recycle the NAD plus, yes? So you have to convert the NADH back to NAD plus. So that is where the reduction of pyruvate to lactate occurs. So here, when pyruvate is reduced to lactate, the NADH is oxidized to NAD plus. NAD plus is regenerated, yes? So now this NAD plus can go back to the reaction 6 of glycolysis, so that ATP production can continue occurring. Does that make sense? Yes. Sorry? I'm so sorry, I can't hear you. Why can't NADH? Why can't NADH used as an electron carrier? No, because so NADH already has electrons. It cannot accept any more electrons. It is already in the reduced form, so it can't accept any more electrons. NAD plus, the positive sign, shows that it has the capacity to accept electrons. Once it accepts the electron, it gets reduced to NADH. It can no longer accept any more electrons. It is already electron rich. Now it can only donate electrons. So NAD plus is an electron acceptor, whereas NADH is an electron donor now. Clear? So that is why NADH cannot participate in the oxidation, because it can only now donate electrons. It cannot accept any more electrons. Clear? Yes? So in under anaerobic condition, in order to generate NAD plus, pyruvate is reduced to lactate, so that NADH is converted to NAD plus. This NAD plus can now move to glycolysis so that ATP production can occur. Clear? In the case of aerobic respiration, whatever NADH is formed, it moves into the mitochondria. It moves into the mitochondria, where in the electron transport chain,
it enters into the electron transport chain and it donates its electrons to the electron transport chain and is converted to NAD+. Then it moves back into the cytosol where it participates in glycolysis. Clear? But in anaerobic glycolysis, it occurs in cells which lacks a mitochondria, isn't it? So because it lacks a mitochondria, there is no electron transport chain occurring there. So you need another mechanism by which NAD+ can be regenerated. And the formation of lactic acid is that reaction by which NAD+ is regenerated. Clear? So NAD+ is regenerated, it can now participate in glycolysis, produce more ATP molecules. So in this way, there is a constant supply of NAD+. Clear? So the formation of lactate is a major fate of glycolysis in anaerobic glycolysis, and this anaerobic glycolysis occurs in cells such as the lens and cornea of the eye, the kidney, medulla, testis, leukocytes, red blood cells, because all these tissues or all these cells are poorly vascularized, meaning there's not enough oxygen reaching these cells. Clear? So in these cells, the glycolysis occurs under anaerobic condition because they are poorly vascularized, meaning the blood supply to these cells are, as in there's not enough blood supply to this cell. So meaning oxygen supply to these cells are very limited. Clear? So these cells carry out anaerobic glycolysis to produce ATP molecules. Then lactate formation in muscles. So in exercising skeletal muscles, so when you're doing prolonged muscular activity, the amount of NADH in the muscle increases or it will become high. And in order to again regenerate NAD+, so that a constant glycolysis can occur continuously in the cells, the NAD+ must be regenerated, isn't it? So again, lactic acid formation. So the pyruvate, whatever pyruvate is formed, it is converted to lactate so that NAD+ can be regenerated, and this NAD+ can now rejoin glycolysis so that ATP production can keep occurring. So during prolonged muscular or muscular activity, as more and more glycolysis occurs under anaerobic condition, there is accumulation of lactic acid in the skeletal muscles. Lactic acid, because it is acid, it causes acidification of the skeletal muscles, and this causes muscular cramps. So I'm sure you might have experienced at some point in life when you're doing muscular activity, or if you're running for a very long time, or even like short bursts, if you give it your all and you're running, you suddenly experience severe pain in your muscle. Yes? That is due to the accumulation of lactic acid. Your lactic acid accumulation causes acidification, and that is what causes the muscular cramp. But once you relax, once you stop exercising and allow blood to be supplied to that muscle, the lactic acid is removed from the skeletal muscles, and the cramping effect or the pain is relieved. Yes? So yes, so muscular continued or sustained muscular activity causes muscular cramp due to the accumulation of lactic acid, which causes acidification of the muscle. Lactic acidosis is another condition. We're almost to the end. So this is, yeah, I'm almost done. I can hear you. So we're almost done. No, we still have 30 minutes left. So we shouldn't be complaining. Okay, so lactic acidosis. So elevated concentration of lactic acid in the plasma is termed as lactic acidosis. So when there is high levels of lactic acid in the blood, that is known as lactic acidosis. And this usually occurs when there is a collapse in the circulatory system. So collapse of circulatory system occurs during myocardial infarction, pulmonary embolism, and uncontrolled hemorrhage, when an individual goes into shock. So under these conditions, your circulatory system is your circulatory system collapses. So meaning adequate amount of oxygen does not reach all parts of your body. Clear? Yes? So when your circulatory system collapses, there is inadequate amount of oxygen reaching your various tissues. So then these tissues carry out anaerobic glycolysis to produce ATP molecules. And you know, the end product of anaerobic glycolysis is lactic acid. So as more and more anaerobic glycolysis occurs, more and more lactic acid is formed and it causes lactic acidosis. Clear? So failure to bring adequate amounts of oxygen to the tissue results in impaired oxidative phosphorylation and decreased ATP synthesis. So in order to survive, now the cells carry out anaerobic glycolysis to produce ATP molecule, thereby producing lactic acid. Now the excess amount of oxygen which is required to recover from a period when the availability of oxygen has been inadequate is known as oxygen death. So the excess amount of oxygen that you need to supply to the body in order to recover from the damage that is caused during a period when there was inadequate supply of oxygen is known as oxygen death. Is that clear? Yes? So this is what happens when someone undergoes myocardial infarction or pulmonary embolism. What do you do to such individuals? You put them on oxygen, isn't it? Yes? You give them oxygen, yes? This is in order to supply them the excess oxygen that is required in order to recover from the time when the body did not have. That is known as oxygen debt. Clear? Yes? So in many clinical situations, measuring the levels or measuring the level of lactic acid in the blood allows for rapid and early detection of oxygen debt in patients and monitoring the recovery. So meaning, if you know a person has undergone, if you know a person has undergone myocardial infarction or some form of pulmonary embolism, they are on oxygen, you have put them on oxygen. Now you keep monitoring their blood lactic acid levels. If their blood lactic acid levels are dropping, that means the amount of oxygen that you're giving them is enough. Like over a period of time, their blood lactic acid levels will be normalized as well as their oxygen levels will also be normalized. But if their lactic acid levels are still very high, that means you need to give them more oxygen. Clear? Meaning, because if the lactic acid is still increasing, that means their body is still doing anaerobic glycolysis, yes? Meaning the tissues are still not receiving enough oxygen. So then now you need to increase the supply. Oh, you want to find one, okay. Yes. Please, we're not done. I said if there's any question, you still see me standing here, isn't it? If I were done, I would have walked out. I wouldn't just be sticking around here. So yes, we're not done yet. I still have like 15 minutes and there are people who need to ask questions. Unless you don't care about the questions and you know everything, you have no doubt. Yes. Then this will be out with this. Oh, the Glyceraldehyde phosphate dehydrogenase. Oh, the poly-ADP ribose polymerase. Yes. So, yes. So it's basically so poly-ADP, that enzyme, it adds ADP ribose to different types of proteins. So those different proteins have other functions in the body, but one of the function is again addition of ADP ribose to this causes inhibition. So yeah, it's just one of its functions. Any other question? Yes. Yes. So your body only produces enough of this amount. It does not keep producing as and when. So, like I said, only a limited amount of NAD plus will be produced, and it is recycled in the body. But once it reaches its lifespan, it will be degraded and new NAD plus will be formed. But in a given in a given time, only a particular limited amount of NAD plus is synthesized and stored in the body. So whatever reaction uses it, they will need to recycle it. Meaning if all the NAD plus has been converted to NADH, unless the NADH is converted back to NAD plus, the reaction will not take place. So it will sort of become like an inhibitory effect. So that is why, in order to maintain a constant supply of NAD plus, your body or your cell has different systems to regenerate it. Under aerobic condition or in cells that have mitochondria, whatever NAD plus is converted to NADH, all that NADH will move into mitochondria, where it will give its electrons to electron transport to form NAD plus. And it now moves back into the cytosol, where it can again participate in glycolysis. So just because all the NAD plus has been converted to NADH does not mean that now the body will start producing NAD plus. No. You will have to convert the NADH back to NAD plus in order for that reaction to take place. So it is sort of cyclic. At a given point, only a limited amount of the molecule will be present in the cell. You can't keep, your cell will not keep synthesizing more and more of it. Yes. Sorry? Sorry? The review for the test so that they can revise form. That's why you have your notes. So basically, are you just going to go through your test and that's your revision? Is that no? That's why you're given notes. We do not give you the test for reviewing so that you can go through. No, you go through the notes. That's what you do. Yes, that's how it's done. Like in med school, that's how we do it. Now you know. You need to work. Listen, that's another thing. In med school, you don't get hand. So yes, we do give you your lecture notes and whatnot, but you are expected to read on your own. Because in this 2 hour or in this 1.5 hours, I cannot discuss everything that is there about glycolysis. I can only give you the important information. Based on that important information, now it's up to you to read further. I mean, ask any meds. You know what you need to do? Come to Ridgeway, ask your seniors how about biochemistry, how you need to learn and how you need to do extra reading. People, what they do is we give them the lecture notes and they read on their own. They make the effort to read on their own and get extra information. You can't just learn what's given in the lecture note. No, that's just a basic, sort of like a foundation, and it's up to you to build on that foundation. Because if I want to teach you about glycolysis, I'll probably need like 2-3 sessions to teach you everything there is about glycolysis. But instead, giving you the foundation, and it's up to you to now read. So doing this review of the test, no, those are shortcuts. No, we're not going to entertain that. So please put in the hard work. You are in med school. If you have decided to become doctors, you should be willing to put in the hard work. Yes. You mean here? Okay, so this proton is you have protons and ions present in the cytosol. So yes, the proton is already present. So you have all sorts of inorganic solvents and anions and cations present in these cytosols. So that's where the protons are available from. So as long as it's not specified, then it doesn't mean there are any particular coenzymes that are required. If the reaction, so like in this case, so like here you have Mg2 plus that is mentioned, isn't it? So meaning that is a cofactor that the enzyme requires in order for it to be fully functional. So if a coenzyme or cofactor is required, then it will be mentioned in the reaction. Or else, not all enzymes require coenzymes or cofactors. Sorry, in preparatory phase, what? The fifth reaction. Yes. They're reversible. Yes, because here dihydroxyacetone phosphate and glyceraldehyde phosphate are isomers of each other. They can be converted to each form by the same enzyme. So it's a reversible reaction. So the only irreversible reactions in glycolysis are reaction 1, reaction 3 and reaction 10. These are the only 3 reactions which are irreversible. The remaining 7 reactions are reversible, meaning the same enzyme can catalyze the forward as well as the reaction in the forward as well as backward direction. So here, so as I mentioned, dihydroxyacetone phosphate, it cannot participate or it cannot continue in the glycolytic pathway because there's no enzyme that can act on it. So now the only other choice is for it to be converted to glyceraldehyde. So as more and more dihydroxyacetone phosphate is produced in the cytosol, it favors its conversion to glyceraldehyde 3-phosphate, so that glyceraldehyde phosphate can be utilized by the glycolytic enzymes to produce energy. Or else, otherwise, dihydroxyacetone phosphate has no other. It can be converted into glycerol 3-phosphate, which is again required for the formation of triacylglycerol. But again, it depends on the metabolic needs of the body. At this particular point, when glycolysis is occurring, the metabolic need of the body is to produce ATP molecules. So all these molecules, they will be channeled into the production of ATP. So here, dihydroxyacetone phosphate, it is converted to glyceraldehyde 3-phosphate so that more ATP molecules can be produced. But if it wasn't, let me not confuse you, let me just stop you there. Yeah, so that's what happens. Yes. They have low blood sugar, as in low blood glucose, and they have. Okay, so your GLUT3, it is not affected by the presence of insulin or glucagon. They are active there. As long as glucose is available in the system, it will be taken up by the GLUT3. Now, glucagon, it is secreted when your blood glucose levels are low, isn't it? So glucagon will now trigger or it will activate those pathways which will generate generate those molecules such as the breakdown of glycogen, or another pathway which is known as gluconeogenesis, which we will learn later. So gluconeogenesis is a pathway where glucose molecules is synthesized from non-carbohydrate molecules like lactate, pyruvate, glycerol. All these molecules can be used to synthesize glucose. So glucagon will activate gluconeogenesis so that more and more glucose is produced. So this glucose can then be utilized by your brain cells as well as your RBCs. So that's why I said glucagon, it favors those pathways which generate glucose, whereas insulin, it favors those pathways which utilizes glucose. Clear? Yes? So because insulin's function is to lower blood glucose levels, so it will activate those pathways which utilize glucose, meaning remove glucose from circulation. Whereas glucagon, its function is to increase blood glucose level, so it will activate those pathways which synthesize and release glucose into the blood. Hope that's clear. Yes? Sorry? Sorry? Why is it using inorganic phosphate instead of organic phosphate? Inorganic phosphate is the ion that is present in the cytosol, the form. So it's easy for this enzyme to. So again, it depends on the enzyme what it what its active site can uses as a substrate. So glyceraldehyde triphosphate dehydrogenase, it uses glyceraldehyde triphosphate as well as inorganic phosphate as a substrate to carry out this reaction. All right. If there's no other questions. All right. Thank you, everyone. Also, it's Friday, so enjoy your weekend.